I'm Bryan Rosado from the Inter-American University of Puerto Rico, and I was working with the role of S-adenosyl methionine synthase in nuclear attraction. We just call it SAMS. So most plant viruses need insect vectors to transmit the virus, and also viruses affect this by maybe changing the volatiles that the plants are emitting to attract these aphids and mediate this propagation. Um, there is some research from a lab that showed that ethylene signaling is very important for aphid attraction, and other research showed that terpene synthase is also attracting more aphids to those plants. And but we don't really know if those are connected in any way. So I did some choice tests where I treated infiltrated SAMs in some plants and GFP as a control in others, and drop some aphids in the middle of the cage, let them choose overnight. And what we saw here was that there was not any significant differences between the two treatments. We wanted to understand a bit more about why was this happening. So we did some RT-qPCRs, first for the terpene synthase levels, because we know that attract aphids, but we still are not sure if that's related to SAMS in any way. We found out that SAMS is actually upregulating the transcript level of terpene synthase, and that's not what we see in the choice test. We wanted to see. We were wondering why, so we went for the defense. Maybe the defense of the plants is what making aphids not choose this plant more. But what we see is the contrary. SAMS is actually downregulating the transcript levels of plandipensine. So we're getting different results in the choices versus the RT-PCR, and we are not sure about why. Maybe the overexpression of SAMS is causing other physiological changes that aphids don't like, or maybe there could be other enzymes related to the pathway that we're not aware of. Maybe other defense that we're not testing here. Either way, understanding more about SAMS could help develop better strategies against pest management.
Thank you. Very nice. What would you do differently? How would you set up your bioassays differently in the future to kind of tease out this contradiction in results you're seeing?
I don't think I would change anything about the assay because that's just data. But what I could do is do more tests, maybe more RT-qPCR to see how more defense genes relate to this pathway, even if there's no literature connected to the ethylene signaling. We didn't know if TPS1 was related to this in any way, and we showed that it was connected to SAMS. So yeah, more RT-qPCR.
Very nice. Thank you so much for sharing your work with us.